Our next speaker, so Chris, could you unshare your, yeah. Our next speaker is Eva Nogales from Berkeley and HHMI. I think her talk is recorded, but she's actually online with us so that she'll be able to answer questions. So with that, I would ask that we show Ava's pre-recorded talk.
Thank you very much to the organizers for making me part of this celebration of the Protein Data Bank. Today, I would like to talk to you about our studies of two human large transcriptional coactivator complexes, TFIID and SAGA, and how they are related both in function and structure. My lab has been characterizing the POL2 transcription pre-initiation complex for a number of years. This is a large assembly that comes together at the core promoter of POL2 genes to position the polymerase with respect to the start site and open the duplex DNA for it to be able to act on. It starts the whole process, is thought, with the recognition of core promoter sequences by TFIID, which is a very large complex itself, 1.3 megadaltons, and contains the TATA binding protein and about 13 TBP associated factors or TAFs. In addition to recognizing core promoters, the complex then recruits the rest of the transcriptional machinery. And not only does it play this general role, but it can also control the amount of transcriptional output through its interaction with gene-specific activators and repressors, as well as chromatin marks. So, however, TFIID have proven very difficult to characterize structurally. It is very large, so it's non-trivial to reconstitute it for overexpression from our expression system. We so far have immunopurified it from HeLa cell, and there are very few copies of the complex, so very scarce. Additionally, the complex is not very stable, so we have to go out of the way to keep it happy and functional. And finally, it suffers for very dramatic flexibility. And this is something that we found out very early on as we started working on it by cryo-EM, but it became very obvious when Mike Cianfrocco, when he was a graduate student in my lab, now he has his own at University of Michigan, showed that of the three lobes that make the complex, lobe A moves very dramatically with respect to a more stable BC core, and that movement is of the order of 150 angstrom. So looking at the population present at equilibrium, the complex changes from a canonical state in which lobe A interacts with lobe C to a rearranged state in which lobe A interacts with lobe B. And which is what was really exciting is when Mike added core promoter DNA and TF2A, which is known to facilitate the engagement of TF2D with DNA. He saw this shift in the population towards the rearranged state. And when he sorted the state, it was only this rearranged state that bound to DNA. So this pointed out to the relevance of the flexibility in the engagement of TF2D with DNA, and also hinted that regulatory factors could act by changing the occupancy of different states in TF2D and therefore tuning the capacity that the complex has for binding DNA. That was before direct detectors, so before the resolution revolution. Once we had a detector, Robert Lauder, another graduate student, decided to use it to study TF2D that was stably bound to DNA and to A. And he did this with biotinylated DNA bound to a streptavidin coated bits for his incubation using small volumes that then he could elute after washing using a restriction enzyme. And compared to what Mike had obtained previously, he improved the structure and showed very clearly that of lobe A, the one that we saw moving, the yellow part, only a segment is present in a stable way bound to DNA in this structure, while the rest is very flexible and in fact inhibiting us from pushing the resolution. So we mastered and do focus refinement to improve the resolution to the point that Robert could take the crystal structure of TBP2A and the TATA box and dock it into the cryo-EM map at one end of the DNA, and then just moving along the length, position the other core promoter sequences that we use in this study, allowing us to see that indeed they're being touched, they're being recognized by density corresponding to protein. And we were able to further interpret the map by doing further focus classification, refinement, improving the resolution to about 7 angstroms, at which point Robert could dock the crystal structure of TAF1 and TAF7 and homology models for TAF2 and 2 copies of the heat repeat of TAF6. And even a little segment, we were brave enough of TAF8 that we thought was part of that interface. So we were left now with a structure that told us what elements, especially TAF1 and also TAF2, are involved in recognizing downstream core promoter sequences. By the way, the INR include the transcription start site, the plus one. But we, of course, wanted to also have the structure of lobe A and lobe B, and this was accomplished by Abhi, another graduate student, Abhi Patel, who's now a postdoc at Northwestern University. He collected a massive data set of the APO complex, TFIID by itself, and he was able to describe better the conformational landscape of lobe A, but also obtain a consensus structure for the BC core, shown in blue at about 4 angstrom resolution, and for lobe A at about 9 angstrom. And using this hybrid model, he could assign subunits to all the densities and come out with the model that I'm going to describe here right now. And this was possible due to the fact that lobe A and lobe B are related. So lobe B is made up by 3 pairs of histone fold containing subunits, TAF6 pairing with TAF9, TAF4 pairing with TAF12, and TAF8 pairing with TAF10. And they form a hexamer that then buckles against the TAF5WD40 domain. This subunit also contains an N-terminal all alpha helical domain. So this module repeats in lobe A with some differences. TAF8 exists in a single copy and is the one that tethers the complex to the BC, the rest of the BC core. Instead of TAF8, lobe A has TAF13, which then interacts with a 4th pair of histone fold containing subunits, TAF13 and 11, which interact with TBP. And TBP is also engaged by N-terminal segments of the TAF1 subunit, being completely inhibited in the context of TFIID. Now, this histone fold had been predicted and even described crystallographically, and TFIID is a DNA binding complex, so it had been proposed that they would act as histone wrapping DNA, but that's not the case. So this is the histone core, just colored so that you can find the similarities in a structure with lobe A and lobe B. However, if you displace the charge on the surface, you will see that the TAFs are completely missing the positive patches that are involved in interacting with the phosphate backbone in nucleosomes. So this is the structure. Just to remind you, lobe B is attached more rigidly to the into the BC core through this TAF8 subunit. Lobe A is more flexibly attached. It doesn't detach. It is actually connected through 2 flexible linkers, one between the histone fold and the heat repeat of TAF6, and the other one between these N-terminal inhibitory domains bound to TBP and the rest of TAF1. So once we established this organization within TFIID, we decided to go back to the dynamics of the complex and their relevance for DNA binding. And we studied a mixed data set. We were just, again, mixing together 2D, 2D, and the core promoter. And we see a states of the APO complex where lobe A, which now marks, has TBP marked in red, moves between touching lobe A and lobe C. And in these complexes, TBP is completely inhibited by binding to different TAFs. Then we have complexes that are in what we call the scanning state, where the downstream DNA has been already engaged by TAF1 and TAF2, and the DNA now is positioned, and lobe A is positioned so as to be as TBP to be able to scan the upstream DNA. This is a situation in which we think the arrangement, the geometrical arrangement, the way TF2D holds the DNA and the way it promotes the reaching out of lobe A will facilitate the DNA competition with TAF1 elements for the binding to the concave surface. However, the DNA is still not fully bound, is not bent, and TBP must be just touching and going quite often. The rearranged study is very similar, but it has incorporated TF2A, which interacts with lobe B, and therefore gets positioned such that in the presence of DNA and lobe A, it will engage TBP right where it should be for engagement with DNA. And just again, the shear geometry and the local concentration of TF2A would allow TF2A to compete off the regulatory regions and bind to the side of TBP. And this will ultimately facilitate the final binding of TBP, which come also with the release due to aesthetic hindrance of its interaction with TAF11 and 13, popping it off lobe A, and now freeing the surface of interaction of TBP with TF2B, which then in turns interact with polymerase and brings it to the promoter. So we summarize this in an animation with Janeri Wasa, where we also incorporate information on modules at the end of linkers that are involved in recognizing modification place on the +1 nucleotome near the core promoter that will contribute to the recruitment and then the readout of the downstream elements by lobe C, positioning lobe A where it can start scanning the DNA, and then with the help of TF2A, TBP will be able to do its final engagement with the DNA, bend the DNA, and free itself of lobe A, opening the surface for interaction with TF2B, which then will bring the polymerase to the site. So this is the summary, and I just want to reiterate that this process, this very dynamic process, builds on two modules, uses two modules that are based on histone fold containing elements that are used as scaffolds to do two completely different functions in lobe A and lobe B. And I want to emphasize them because these are modules that are shared with yet another cofactor in transcription, coactivator in transcription, which is SAGA, the SPT ADA GCN5 complex. This is a complex that, like TF2D, is present at core promoters. It gets there especially through the interaction with gene-specific activators through its very large trap subunit, which is engaged by a core of TAF-containing elements and histone fold-containing elements that binds the TBP. And in addition, it has two histone modification modules, a histone acetyltransferase and a histone deubiquitinase. So again, building on this core, a scaffolding core of histone fold-containing subunits. So the structure of yeast saga from two different species was reported last year from the labs of Patrick Kramer and Patrick Schulz. The TRAP1 subunit with the homologue of the human trap and the core module were defined at resolution between 3.5 and 3.9 angstrom. The HAT and DAP were very flexible and were only seen at much lower resolutions. Human, however, is quite different from yeast. The sequence conservation is very poor, but most importantly, there are numerous deletions, there are insertions, there are gene duplications that happen in human for some of the types that do not happen in yeast. There's a lack of one of the yeast subunits, the SPT8, which is involved, contributes to the binding of TBP. However, humans have an additional splicing module. And while the complex is not essentially in yeast, it is absolutely required for metazoan embryonic development. So we went for the human complex. We purified these from HeLa cells. Actually, Megan did by introducing a tag using CRISPR in the chromosomal copy of one of the subunits. So this is the endogenous material, again, from human cells. And then Dominic Herbst, a postdoc in my lab, carried out the cryo-EM analysis. And he showed again that the DAP and HAT modules are very flexible, in fact, even more flexible than in yeast, and we barely detect them in 2D class averages. The splicing module is attached to the heat repeat of the TAP6-like protein, and that is flexible, and we only see again at low resolution about 19 angstrom. The rest of the complex we see at an average of an overall 2.9 angstrom resolution. This is the local map. So we were able to generate a model, either doing docking of the crystal structure for the splicing module and a homology model for this heat repeat, or building and refining directly into the density for the rest of the structure. Okay, so I'm going to tell you first that the module, and I'm always going to use this color coding to refer to the different elements. Again, is built on an octamer or histone fold containing subunits, and which are explicitly indicated here, that then dock against the WD40 domain of the TAF-like subunit in human SAGA, distorting that histone-like core with respect to what is present in a nucleosome. Now, the N-terminal domain of this TAF5-like protein is very interesting, is engaged in interactions with another subunit in the core, the SAPT20H. And this interaction actually does not exist in budding yeast. So the module is instead have moved by 60 degrees and is interacting with the heat repeat of TAF6 right at the side, which in the human complex incorporates the splicing module. So differences in those two elements can give rise to an interaction or not that ultimately result in the incorporation or not of a splicing module. So I will continue to talk about the SAPT20H subunit. That is the N-terminus that interact with the N-terminus of the TAF5L. But the subunit is much larger in human. It has an extension that wraps around the large trap subunit through and then ends up with a C-terminal domain. Now that is very different in yeast. So here I'm showing you a different view where I emphasize, in addition to the yellow subunit, the TAF12 subunit in cyan. And then I compare to the yeast structure in which the SPT20 subunit ends right there. So it's like in the whole latch and C-terminal domain. But on the other hand, it has a TAF12 that is longer. It has an extension at the N-terminus, and it also wraps about the TRA1 subunit, but in a different direction, ending with this N-terminus right where the C-terminus terminal domain of the yellow subunit is in human. Now, the whole contact of the core with the TRA subunit is very different. Yeast has a cleft so that only a couple of subunits are actually involved in the interaction, while this is a very busy interface where almost every single subunit is making contact with the TRA. And furthermore, that interaction is so very different in nature that if you superimpose the core complex, the TRA subunit rotates by more than 70 degrees, meaning that different functional elements in the core and TRA subunit are in different geometries in yeast and humans. So going back to the T20H C-terminal domain, this displays at the exit of a tunnel that at its other end has a density that we have been able to identify as a phosphorinositides at IP6. And it sits in a region that is very highly conserved, very positively charged, and was not seen probably due to the resolution in the yeast structures. Now, the TRAP subunit is as I told you, is the subunit that interact with gene specific activators and repressors, including some important oncogenes like MYC. And we can now map those regions in the structure and put it in the context of human mutations. Some important in cancer, like the S721 to phenylalanine in melanoma, but also others that are involved in neurological development in humans. So we can see how some of them are exposed, some of them are buried and will contribute to the conformation of the site. But I just intrigued by two single point mutations present in neurological diseases, in genetic diseases in humans that are involved in interactions of TRA1 with the sub-20H at two regions, the latch and the C-terminal domain, that do not exist in yeast, where the protein finishes right here, underscoring the relevance of human structures when we want to understand human disease. So I want to thank Jay, who over the years have purified TF2D from human cells, from HeLa, Dan Toso, who runs our CalCryo facility, Patricia, who started the cryo-EM studies that showed that TF2D was very flexible, and then Mike, Robert, and Avi, three sequential biophysics graduate students that work on TF2D, and Dominic and Megan for their work on human SAGA. And Megan is a graduate student in the lab of my colleagues Xavier and Tej. And thank you very much for your attention.
Thank you very much. That was a really terrific talk. I learned a lot from it. I was wondering, are there questions from the audience? Either put it in chat. I think, Eva, are you there now? Are you live? Oh, good.
I am here.
Okay.
Helen, may I jump in?
Yes, sure.
Sure.
Please.
So I don't have a question, but Eva, I wanted to thank you for contributing on behalf of the RCSP Protein Data Bank. And I also wanted to tell you what a thrill it was for me to see the structure of the tartar box binding protein bound to DNA in the middle of your complex, and also the structures of the TAF, the histone-like TAFs that we determined subsequently also playing a role in the architecture of TF2D. It's an amazing accomplishment that you and your colleagues made on unraveling what TF2D looks like and how it's working, and I think the video that you produced in collaboration with Janet Awasa is incredibly elegant. Thank you.
Yes, thank you very much for that. Yeah.
We stand in the shoulders of Jan like you. So
I think giant is an exaggeration, but we all benefit from all the work that was done before us, particularly the 180,000 plus structures in the PDB. Okay.
Are there any other questions that anyone would like to ask, or comments? I'm reflecting on 10 years ago with the PDB40 and looking at the science that has happened in the last 10 years. And it's remarkable to see all this. So, Greg has a question. Greg?
Yes, I may as well turn my video on to ask it. You mentioned splicing activity in the SAGA complex. Is there a different rearrangement of subunits when RNA is encountered?
Sorry, said splice activity by splicing subunits. It is still a mystery what those splicing subunits that are shared with the true splicing machinery are doing in there. They are in one and not in the other because basically the surface of interface with the heat repeat of the TAP6L is the surface where those two subunits incorporate into a larger, the larger splicing, the U1. So we are trying to see whether we can see it engage with nucleic acid. But to be completely honest, right now it is a mystery. It is not clear whether it exists in every single complex, but we tagged another subunit, one of the subunits in the core, and what we got is what you see in the structure. So those subunits are present, and what they're doing is still unknown. Yeah, we'll see what we can do in terms of visualizing it bound to nucleic acid. Yeah.